You learn how fatty acids are transported into the mitochondrial matrix via the acyl-carnitine shuttle. In mitochondria, fatty acid molecules are broken down to produce energy. This process, called beta oxidation, can be divided into 4 sequential reactions. In this video, we'll take a closer look at these reactions and at the fate of the end products of beta oxidation. The 4 steps of beta oxidation are essentially the reverse of the 4 steps in fatty acid synthesis. In beta oxidation, the steps are oxidation, hydration, oxidation again, and thiolysis. Each round of 4 reactions results in a shortening of the fatty acid by 2 carbons. The first step is an oxidation that introduces a double bond into an acyl coenzyme A, forming a trans-enoyl coenzyme A. In the structure shown here, the letter R indicates the remaining of the fatty acid chain. The oxidation occurs at the beta carbon, hence the name of beta oxidation. The reaction is catalyzed by the acyl coenzyme A dehydrogenase and is coupled with the reduction of FAD to FADH2. FADH2 can then transfer its electrons and protons to the electron transport chain. Note that the term delta 2 in the name trans-delta 2 enoyl coenzyme A means that the reaction product has a double bond at carbon 2. The second step is the hydration of the double bond in enoyl coenzyme A to form L3 hydroxyacyl coenzyme A. This reaction is catalyzed by the enoyl coenzyme A hydratase and eliminates the double bond. The third step is the second oxidation in the process, resulting in a formation of 3 ketoacyl coenzyme A. This time the oxidation is coupled with a reduction of NAD+ to NADH and H+. Once again, NADH will feed the electron transport chain. In the last and 4th step, the enzyme called beta-ketothiolase catalyzes the cleavage of the 3 ketoacyl coenzyme A into 2 molecules at the site indicated by the blue arrow. The thiol group of an incoming acetyl coenzyme A is involved in a cleavage. The result is the release of a molecule of acetyl coenzyme A and the attachment of the remaining fatty acid shortened by 2 carbon to the incoming acetyl coenzyme A molecule. This shorter acyl coenzyme A can now undergo a new cycle of beta oxidation. How much energy is produced by the complete oxidation of a fatty acid molecule? Let's take the example of a 16-carbon fatty acid, palmitate, starting with palmitoyl coenzyme A. 7 rounds of oxidation will be necessary to release 8 molecules of acetyl coenzyme A, 7 molecules of FADH2, and 7 molecules of NADH. Each acetyl coenzyme A entering the citric acid cycle will yield 10 ATP. So if the 8 molecules of acetyl coenzyme A that are released enter the citric acid cycle, a total of 80 molecules of ATP will be produced per molecule of palmitoyl coenzyme A. Each FADH2 entering the electron transport chain will yield 1.5 molecules of ATP. So if the 7 FADH2 molecules enter the electron transport chain, a total of 10.5 molecules of ATP will be produced per molecule of palmitoyl coenzyme A. Finally, each NADH molecule entering the electron transport chain will yield 2.5 molecules of ATP. So if the 7 NADH molecules produced by the oxidation of palmitoyl coenzyme A enter the electron transport chain, an additional 17.5 molecules of ATP will be produced. So overall, the degradation of one palmitoyl coenzyme A releases 108 molecules of ATP. But as you recall, 2 ATP are needed to convert palmitate to palmitoyl coenzyme A. So the net production of ATP is 106 molecules per molecule of palmitate.
What happens with fatty acids that have an odd number of carbons?
The beta oxidation for these fatty acids is largely the same as described before. However, the final round of beta oxidation yields one acetyl coenzyme A and one 3-carbon propionyl coenzyme A, which is converted to succinyl coenzyme A to fuel the citric acid cycle. You could also ask, what about unsaturated fatty acid? How they are broken down? Additional steps are needed to oxidize unsaturated fatty acids. Let's take the example of a 16-carbon fatty acid with a double bond beginning at carbon 9. The first 3 rounds of beta oxidations are normal. At this point, the double bond is reached and a molecule of cis-enoyl coenzyme A is produced. The double bond is on the 3rd carbon relative to the carbonyl group. The next round of beta oxidation cannot proceed because cis-enoyl coenzyme A is not a substrate for the dehydrogenase. The normal substrate is acyl coenzyme A, which is converted to trans-enoyl coenzyme A during step 1 of the beta oxidation cycle. Here, before beta oxidation can resume, an isomerase converts the cis-enoyl coenzyme A to trans-enoyl coenzyme A. Upon conversion, a new round of beta oxidation can start at step 2 instead of step 1 of the beta oxidation cycle. So our problem is solved. What happens if the double bond is at carbon 4 relative to the carbonyl group? Yet another additional step is needed. During step 1 of the cycle, the acyl coenzyme A dehydrogenase will insert a second double bond at carbon 2. Then a reductase uses NADPH to bring the number of double bonds back to 1. Finally, an isomerase shifts the double bond to form a trans-enoyl coenzyme A. The shift results in the production of the substrate for step 2 in the beta oxidation cycle. So beta oxidation can now resume. In summary, fatty acid synthesis and beta oxidation happen in distinct cellular locations: synthesis in the cytoplasm, beta oxidation in the mitochondrial matrix. Malonyl coenzyme A plays an important role in both processes. It promotes fatty acid synthesis by providing the needed 3-carbon blocks. It inhibits beta oxidation by repressing CPT1 and blocking the acyl carnitine shuttle. Finally, both processes involve multiple rounds of 4 sequential reactions, which add during synthesis or remove during beta oxidation 2 carbon groups at a time. So now, what is the fate of acetyl coenzyme A produced by beta oxidation? We've already discussed the fate under normal conditions. Acetyl coenzyme A feeds the citric acid cycle by reacting with oxaloacetate to produce citrate. Acetyl coenzyme A will thus contribute to ATP synthesis. If the supply of oxaloacetate is low, during starvation or in people suffering from diabetes, the acetyl coenzyme A accumulates and is converted to ketone bodies. The presence of ketone bodies is an indicator of an active breakdown of fat and a depleted carbohydrate supply. Where and how ketone bodies are formed? The liver is the main source of ketone body synthesis, and this synthesis will take place in mitochondria. 3 molecules of acetyl coenzyme A react to form the ketone acetoacetate and regenerate 1 acetyl coenzyme A that can be used again to make more ketone bodies. If the concentration of NADH is high enough, acetoacetate is reduced to form D3-hydroxybutyrate. Acetoacetate can also spontaneously and slowly be decarboxylated to release acetone. Acetone can often be smelled on the breath of a person producing ketone bodies. So what are ketone bodies used for? They are distributed to tissues such as heart and kidney, where they serve as a source of fuel molecules. While the brain prefers glucose, it can also metabolize ketone bodies during starvation and in diabetic patients. The 3-hydroxybutyrate is converted back to oxaloacetate. NADH is produced and can feed the electron transport chain and thus contribute to the formation of ATP. Acetoacetate is then converted to 2-acetyl coenzyme A and 1-succinyl coenzyme A. Both molecules can feed the citric acid cycle as long as tissues have enough oxaloacetate.